Dear students, let's discuss a very important concept, that is the concept of Huntington's chorea. Huntington's chorea is basically a single gene disorder. It is a dominant single gene disorder related with a gene which is present upon the chromosome number 4. So, this is the graphical representation of the chromosome number 4, and you see on this chromosome number 4 there is a gene, this gene. This gene is known as HTT gene. HTT stands for Huntington's gene. This gene was basically discovered in 1993, and this gene, which you see upon the chromosome number 4 in case of human genome, if it undergoes mutation, it leads to a genetic disorder which we call as Huntington's chorea. Now, before discussing in detail about the Huntington's chorea, let me first of all discuss the normal functions of this gene. That's the Huntington's gene. So again, I will repeat this thing, that this gene was reported by some geneticist in 1993. Now, what is basically the function of this gene? This gene is of enormous importance with respect to our nerve cells. It produces a protein which we call as Huntington's protein, which is of enormous significance in case of our nerve cells. This gene, when it completes its central dogma, that's when it first of all transcribes into the messenger RNA and later on translates into the proteins, it will form a protein which we call as Huntington's protein, or we simply say as HTT protein. HTT protein stands for Huntington's protein. Now, when you try to analyze the gene sequence of this gene, when you try to analyze the nucleotide sequence of this gene, you will see in this gene a particular sequence of nucleotides, that is C A G C A G C A G. This sequence of nucleotides is repeated in this gene for at least 20 times. What do you mean by that? It means there is a nucleotide having the nitrogenous base cytosine, followed by another nucleotide having the nitrogenous base adenine, followed by the third nucleotide having the nitrogenous base guanine. And this sequence is repeated over and again. Again, there will be a nucleotide with the nitrogenous base cytosine, then a nucleotide with nitrogenous base adenine, then a nucleotide with nitrogenous base guanine. And this sequence of CAG nucleotides is repeated at least 20 times in this Huntington gene. This is the normal sequence of the gene. And now try to understand the normal functions of this gene and the normal functions of the gene product of this gene. That's the Huntington protein. Once it completes the central dogma, this gene completes the central dogma, means it completes the transcription and translation, it will form a protein that is known as HTT protein. Now, you will see, if I if I draw the neuron, this is the cyton of the neuron, and this is the nucleus of the neuron. When we try to understand the functions of this Huntington protein, you will see, once the Huntington protein is formed, I will graphically represent the Huntington protein like this. Once the Huntington protein is formed, it directly goes into the nerve cells or into the neurons. But remember over here, this HTT protein has a tail which we call as polyglutamine tail. This is the polyglutamine tail of the HTT protein. Now, as the CAG, you see this codon CAG upon messenger RNA, it codes for glutamine. So it is present 20 times in the this HTT gene. So 20 such amino acids will be present in this polyglutamine tail of the HTT protein. And you will see once it is formed, the HTT protein is formed, it goes into the nuclear, it goes into the neurons, and while it has a very important role, particularly with respect to the this nucleus of the neurons, it somehow impacts the command center of the neurons, that's the nuclei of the neurons, to carry out some essential tasks and essential activities which are very necessary for the maintaining the normal health of the neurons. Now, so this is the HTT protein. So once the HTT protein is formed, it will go into the neurons. Once it goes into the neurons, it is responsible for normal functioning of the neurons. So it is the normal condition. Now you will try to analyze, sorry, we will try to analyze what will happen if this HTT gene undergoes mutation. Again, I will say this is HTT gene, dominant HTT gene. It's HTT gene, means Huntington gene. If it undergoes a mutation, you know, mutation is any molecular change in the structure of DNA and the structure of gene. If it undergoes a mutation, in Huntington's chorea, it undergoes a mutation in which you will see I have already explained to you the CAG sequence present in the Huntington gene, and I have already made it clear that this sequence of CAG, it is present at least 20 times in the CAG, in the Huntington gene. Now what happens once it undergoes mutation, you will see the CAG sequence is repeated about 36 times in the Huntington gene. So there is an abnormality. A sequence which needed to be repeated 20 times in usual case, it now gets repeated 36 times. And this creates the problem. You already see that this CAG codon, it codes for glutamine. Once the once the gene undergoes mutation, now it will not form the normal Huntington protein, it will form an abnormal Huntington protein which we call as mutant Huntington protein. So again I will draw the protein, but it is a mutant protein. It's a mutant protein. What is the difference between the normal Huntington protein and mutant Huntington protein? Try to understand over here that in normal Huntington gene, the CAG sequence is repeated only 20 times, but in the this mutant HTT gene, the same sequence CAG, it is repeated 36 times. It will have impact on this polyglutamine tail. The polyglutamine tail of the mutant HTT protein will be, a mutant Huntington protein will be considerably long as compared with the normal HTT protein. Because in the normal HTT protein, you will see in the tail of the protein there would be 20 glutamine amino acids, but in the mutant HTT protein there will be 36 glutamine amino acids in its tail. This is what creates the problem. Again, you will see that I will draw over here the cytosol of the neuron. This is the cytosol, this cytosol, this over here axon, this is the nucleus of the cytosol. The what we call as the mutant HTT protein will now go into the nucleus of the neuron. Once it goes into the nucleus of the neurons, it creates problems. Once the normal HTT protein goes into the it goes into the neurons, it controls the command center, and what we call as it carries out certain functions which are necessary for maintaining normal functioning of the neurons. But when it is mutant HTT protein, it creates problems in the way that once the mutant HTT proteins go inside the this neuron, you will see clumping of these mutant HTT proteins occurs by their long polyglutamine tails. Means all the mutant HTT proteins which go into the nucleus of the neuron or in other parts of the neuron, you will see they get stick to one another, they clump with one another by their long polyglutamine tails. These are the glutamine tails, and by these glutamine tails they get attached to one another, resulting in the what we call as clumping of mutant of mutant HTT protein. So once these mutant HTT proteins form clumps inside the neurons, particularly in the nuclei of the neurons, it begins the uh, process of nerve degeneration and neuron degeneration. They damage the nerve cells. Now these proteins, mutant HTT proteins, turn out to be the toxic proteins. They damage the neurons. They kill the neurons, and this brings about the nerve degeneration. Now, this is not only the single process. You will see these, in fact, these mutant HTT proteins will form clumps, and this will create definitely problems. But the problems are much more increased by another what we call as problem created by the mutant HTT protein. Now the mutant HTT protein, besides forming clumps with other mutant HTT proteins, it will get attached with other proteins which have glutamine, what we call as chains inside them. It's not only these what we call as these HTT proteins which have glutamine tails in them, but there are many other proteins inside the neurons which have glutamine sequences in them, and it corrupts, rather it kidnaps all those proteins which have glutamine these sequences in them. These mutant HTT proteins kidnap all those proteins which have glutamine tails or glutamine chains in them, and prevent them from performing their usual functions. This increases the complexity of the problem, already existing problem due to mutant HTT protein. Because once those proteins are kidnapped which contain the glutamine sequences, you will see it inhibits many other functions which were carried by those proteins inside the neurons. So it will intensify the problem of the nerve degeneration, and it will bring the degeneration of the neurons, rather it will kill the neurons. And you will see now, after that what will happen, you will see, first of all, these neurons, this nerve degeneration will begin in the brain. This is the representation of the portion of the brain. You will see in the brain there are these basal ganglia, basal ganglia, like the globus pallidus and putamen. You will see, first of all, the nerve degeneration will begin in the these basal ganglia, like that of the globus pallidus and putamen. And after that, what will happen? You will see what's the normal function of these basal ganglia. The normal function of the basal ganglia is that they maintain the thought process in our brain. They maintain the sequence of thoughts which emerge or which emanate out from our brain. The thoughts are regulated, or the thoughts emanate in a particular sequence, and this particular sequence of the thoughts which emanates from our brain, it is regulated by the these basal ganglia. The second thing is that it maintains the emotional states of a person. The basal ganglia maintain the emotional states of a person. So the thinking process, the sequence of thoughts, and the emotional processes, they are particularly regulated by the basal ganglia, like globus pallidus and putamen. Now after that, you will see now what will happen. Once the basal ganglia get damaged due to the mutant HTT protein, you will see the thought process will get disturbed in the person, and the person will feel difficulty in regulating the what we call as the thought process in his brain. And the second thing is that there will be emotional disturbances in the brain of that very person. Okay. After that, you will see many other motor areas of the brain will get motor areas. Motor areas of the brain will get affected by the nerve destruction, by the nerve degeneration, and the person will feel difficulty in motor activities of the body, particularly in walking. You will see staggering, clumsy gait in the person suffering from the this what we call as the Huntington's chorea, and the persons will feel difficulty in talking and difficulty in understanding what is being talked to them. Means when another person is talking to them, they fail to understand, or if they understand with difficulty what the other person is talking to them. So they feel understanding in the speech of the other person. Gradually the motor areas of the brain, they get affected, and the person feels much more problems in his body related with the motor functioning of the this brain. Now this disease usually occurs in the age group of 30 to 40, means it occurs in the adults. And once the disease occurs, it is very much difficult to control the degeneration of the neurons. But if this disease occurs before the age of the 20, we call it as juvenile this Huntington's chorea. So if it occurs before the age of 20, which is a rare case, we call it as juvenile Huntington's chorea. uh, but it usually occurs in the age group of 30 to 40, and it becomes very much difficult to control the once the symptoms have onset, once the disease has begun, it becomes very much difficult to control the nerve degeneration. And after the nerve degeneration begins, the patients can live a disturbed life for about 10 to 20 years, after which they die. So this is what happens in the this what we call as Huntington's chorea. Now, lastly, I will discuss about this thing, that is chorea. When you see chorea, chorea means involuntary jerky movements, involuntary jerky movements. As the motor areas of the brain get affected, you will see the person suffering from this disease, they will rather show jerky movements in their body, which are uncontrollable, means involuntary jerky movements in their body. And from that, this has been derived, that is chorea. Chorea means involuntary jerky movements of the what we call as the body. And these are some of the symptoms which are seen in the persons which are suffering from the Huntington's chorea. Thank you.